Hello and welcome again. Today I'll be marking and giving feedback on this biology lab report. This thing here is just to cover the person's name, and I would like to say a big thank you to the student who allowed me to mark this on YouTube. I'll be using a red and green marker. The red one, I'm going to indicate everything that was a problem. The green one is basically all the things that were right. But before I start marking this, I just want to give you a quick overview of what the student actually did as an experiment. Otherwise it might be a little bit difficult. So there is a picture here. I'm just going to explain this. What the student did is the student added yeast, sugar and water into these 3 bottles, and he tried to measure the carbon dioxide production based on the sugar concentration that he added. So that's one thing. And also I think the student measured the height of the foam that was formed. So the hypothesis is the more sugar there is in the higher the sugar concentration, the more CO2 is being produced. Okay, so that is his experiment, and now let's have a look of whether he actually did everything right. So first of all, I can see that the student actually used my template. So he used those headings, and that's already good because this actually indicates that the student actually wanted to complete all of the parts. So I think that's fine. However, on first sight, I think that he did not write a lot. Okay, so it's kind of sketchy. Also over here, it's kind of sketchy what he bullet points. That is not the problem. These points here, they better be really good for the student to actually get the points. So what I will do is I'll quickly skim over the whole thing. I mean, of course, I already read everything, so that it's a little bit faster making this video. And I'm just going to indicate some of the problems here. So a research question is present, but the problem is that the research question is highly unspecific. To what extent do the different amounts of sugar? That is not correct. It's not the amounts of sugar, it's the sugar concentration. The swelling process is not correct. It is not the swelling process. The yeast does not swell. It is the foam formation, okay? And the amount of CO2 produced, that is fine, okay? But the amount of CO2 produced after a certain time, it's of course, the production goes up over time. The amount of sugar is not correct. It's the sugar concentration. The dependent variables is the amount of CO2 produced. No, not the amount. It's the volume. The size of the balloons, well, that is the volume of the balloons, highly unclear. And the size of the grown yeast, the yeast does not grow, it forms foam. Okay, so there is a big issue here already with the research question. Let's move on. Why is this topic significant? I'm doing this experiment to see how the different amounts of yeah, and of course I know that. I mean, he already told me. It's just a repetition. It could be possibly helpful for bakers. How? How? Knowing the effect of the different amounts of sugar might help them prepare better. How? This is not good, okay? There is actually, it's highly unspecific, and background information. Here actually is expected information concerning how yeast respiration depends on substrate concentration, and this is actually not really the background information. He watched some kind of a YouTube video where he basically watch something similar. But the background information, actually I talk about scientific background, and what I kind of hoped to have seen is the following. I'm just going to use this here. This is to actually have the CO2 concentration, how this actually is dependent on substrate concentration, and then we get a curve like this where you have down here the sugar, and here we have the CO2 being produced, and that is basically what I hope to see. There is absolutely no reference to this here. I do not even know why he's telling me all of this. So he just describes the video. That's actually an explanation of how he got the idea of the research question. I mean, I probably would move this here into the front part. Because this kind of explains. I don't know, this could be don't see any personal significance. There are no dangerous chemicals. Yeah, of course. But if you want to get points for this, then I need a little bit more here. Okay, more. The observation method I intend to, so that's basically how do you collect the dependent variable, the amount of CO2 by yeast depending on the amounts of sugar. You already told me that. In order I put 7 grams in different amounts of sugar. Okay, that's already part of the method. Okay, which is fine. I collect the amounts of bottles with balloons. So I can incarcerate. Incarcerate? Are you gonna put the CO2 into jail? Incarcerate means to put into jail. At the end of the experiment, I will compare the volumes. Okay, I think this is as an overview method. That's okay. Okay, in order to make my Okay, so I do expect a more detailed method later on, but I think that's okay. I tell the students always to give an overview method and before they actually jump into details. Yeast to start the experiment. Yeah, water, 3 balloons to incarcerate. I don't like the incarcerate part again. 3 bottles. The size of the bottles is missing. Everything, ruler to measure the radius and the circumference of the balloon. You cannot measure with a ruler. And the height of the grown yeast. No, the yeast does not grow. There are some issues here. So, how do you control the controlled variables, constant variables, the shape of the bottle, how much, what is the size of the bottle, how much water are you going to use, what is the temperature of the water, how are you going to keep it constant, how do you actually, the amount of yeast, of course, is 7 grams, you told me that, okay, you're not telling me how you keep this constant, which type of bottle, okay, which size, yeah, you got to tell me, okay, so this one is definitely more here. It's really important. This is not, it's simply listed, but it does not, actually does not say how he actually controls the variables, how you keep it the same, and at what level you keep it. Exploration, control of the independent variable. So that's basically this measurement of the balloon. Nonsense, not the independent variable, that is the amount of sugar. How do you control this? Okay, I will have 3 bottles and 1 spoon of sugar for bottle. Why spoon? Could you not at least measure the grams? Yeah, or at least the moles per liter. Okay, 2 spoons. Why no control? Where is the control? What about 0 grams of sugar? I don't see that. The control is missing. I will use a knife to take out the extra amount of sugar on the top of the spoon, as I will make sure. Yeah, okay. How do you mix the whole thing? How do you actually? Yeah, that there is more here, and there is. It's a little bit unclear. Yeah. But I mean, yeah. Measurement of the dependent variable, that is now the CO2 production, the foam production. Okay, so let's have a look here. Excuse me, amount of produced CO2. So, there's one thing that we forgot up here. A very important thing I just realized. What about the time? Okay, the time. Yeah, because you have to stop the measurement after a certain time. So how long are you going to actually allow it to ferment? Dependent variables, amount of produced CO2, yeah, size of the balloon, size of the grown yeast. What do you mean? The size of the size of the balloon indicates the amount of CO2. So I don't understand why there are 2 of them here, and this yeast does not grow. I think he talks about the foam production. I will measure the radius and circumference of the balloon. How often? And then use the volume sphere. Okay. I will measure the height of the grown yeast. It doesn't grow. I will compare volumes of the balloons after a couple of hours. That is the lab problem with the control of variables. How many hours to start the experiment that the balloon will be big enough that I can make any. Okay, there's a justification which is fine. Okay, that is okay. Yeah. If I wait longer, the balloons may burst, and if I wait less, I think that's okay. That's a justification. Okay, which is good. But I want to have this type of justification also for the other things, right? So there is really, that there is really more to be written here. So the data processing, how does he process the data after measuring the balloons using a volume? You already told me that. The most CO2. Yeah, oh sure, okay. If you already talk about the formula, what is the formula? More, I will compare this. Yeah, but that's obvious. You got to do this. I mean, these are just empty words. You know, are you going to draw a line of best fit? Are you going to graph something? Okay, all of these things. So here, he presents the raw data. First side, the raw data table looks kind of neat. However, there are some major labeling issues. First of all, each table must have a title, okay, which is missing. I mean, you could say that this here is the title. Measurement on the balloons in centimeters. You know what? I'll take this away. I'll count this as the title here. Okay, that's okay. He's got 3 trials. That already is also okay. He's repeating the experiment, but that's the first time I'm actually seeing that he's repeating the experiment. He's never talked about this before. He should actually mention this in the method. And what's bottle 1, 2, and 3? I have no clue what this is. And where is the sugar concentration here? I don't see the sugar. Of course, he meant the bottle 1, 2, and 3. That's the different sugar concentrations. But you have to say this. Okay, so this is a problem. okay, here as well. So that's one thing. The radius and the circumference of the balloon and the radius and the circumference of the average, that's generally fine. My recommendation is that you actually orient the table in the other direction, that you have the trials here and here the sugar concentration. It's not a big thing. The bigger issue is why are there inconsistent number of significant digits? Here he's got 5.85, then here again, here something is missing, a decimal is missing. So this means an inconsistent number of significant digits or shows that the student is not aware of measurement errors and therefore points are lost. Okay, same problem here. Why not 2.0? I tell you why not, because Excel, the spreadsheet program, takes it away. You have to add them again. Also over here, the average, you cannot have, a calculation cannot have more significant figures than the original values. So this one is too precise. This one lacks precision. This one is again too precise. So that is another thing here, okay? And it's not the grown yeast we talk. I think he measured the height of the foam, and he did not mention this in this method. So what we have here is the following. Basically, he simply uses regular text to describe the table, which is okay. That's actually not only okay, that's what I expect students to do. Okay, he describes simply what he sees, so that is fine. Yeah. So that is many students actually simply present the table and then don't present the data in text form. Okay, that's good. Next one, analysis processing. I don't understand one thing why many students actually, they try to convert a biology lab report into a math report. It's not relevant. Mention in the method how you actually calculate the volume of the CO2. But it is not necessary to actually show the calculations. Some people even show me how to calculate averages. That is not important. Okay, bottle 1, 2, 3, same problem. Volume of the balloons. I'm not interested in the volume of the balloons. I'm actually interested in the volume of CO2 produced. This is actually what we are interested in. So link it to the research question. Same issue here. People spend so much time here showing the math. Just show it to me in the method, how you calculate it. I assume that you can do this stuff, okay? Grown yeast, why did he do that? Well, he compares bottle 1 and 2. Cubic centimeters? I thought he measured the height of the foam. What? I don't understand this. I thought he measured the height of the foam here. I mean, he didn't mention this in the method, but that's what I assumed. So I don't understand this. And I also do not understand the purpose of comparing this. I'm not saying he shouldn't, but why are you doing that? And if you're comparing this, why don't you mention this at least also in the data processing part? Because here you didn't mention it. You bothered to write a separate data processing section here, but then does not actually tell me these things. So I'm completely lost here. The measurement uncertainty, that is important. I mean, he has a paragraph here, but actually is inconsistent about the measurement uncertainty in the tables. Okay, is not significant because I'm interested in the comparison, but that's not the justification. Then you have to compare it. It's not significant because the standard deviation is too small or something like this. But this is not a justification. Yeah. So this is not this is a problem as well. Because you have to actually link it to the research question more. Did I? Okay. So that's basically the problem that we have here. Measurement uncertainty without actually saying how it is relevant. So now we have graphs, okay? The graph shows the volumes. This is, come on, volumes of what? I don't get it. What are the units? I don't get it, okay? So the volume of the balloons, units are missing. What the volume? What in cubic centimeters and liters? Okay, there's some what units missing. This is basically a description of the graph. I think this is. I mean, what I would do is the following. This is some kind of a caption that goes below. I mean that's also some kind of a description of what you actually see. He gave me formulas here. Why did he do that? I'm not saying it's a bad thing. But that's just putting in formulas for the sake of putting in formulas, okay? So what's the point? I'd be more interested really in is there a significant difference That's the thing, right? And he should actually talk about the fact, I mean, these are trials, right? And you can see that over here there is a certain spread of values, which you can see here. That's to be expected, okay? The values go here, all the way from here to here. And this difference here in the values is smaller than the difference between the different independent variables. So there seems to be a significant difference, but he's not talking about this. Okay, rather he gives me some kind of formulas where I do not know why he's giving it this to me. I know why, because the student likes math, and that's why he sees everything through the lens of math, just like biologists see everything through the lens of a biologist, and so on. Economists see everything economic. Oh, I'm drifting off into TOK again, into theory of knowledge. Okay, so that's a different representation. Why, if this is better, why didn't you just give me this? And where are the averages? The averages are missing here. Actually, I want to see the averages. Yeah, that's actually the point. The idea is the following: you can give all of you can actually give the full data in the table, but but the average. These are the things that actually go into the graph. So that's a really volumes totally unclear of what. Yeah, were the units ball 1, 2, 3? I have no idea what this means. Okay. It's actually the different amounts of the sugar concentrations. The size of the grown yeast, as we same. Height, now I know it. Here it's height. So that's actually in centimeters, I suppose. And here he told me that it is in cubic centimeters. Or is this the difference in volume? This could be that he actually wanted to calculate the difference in volume here, not the height. Ah, that's probably the thing. And then cubic centimeters, actually, here he wanted to calculate the cubic centimeters, that's the volume. That's actually the thing, but this is grown yeast. Grown yeast, that was the foam height, right? But now he actually talks about the volume. I'm totally confused. Okay? So, I don't know, this is a contradiction here. And then we move on. Here there is a picture, qualitative data. You got to have qualitative data. It's a picture here. I don't know if what's so qualitative about this. I don't know. I don't understand. What would be kind of interesting is the following. Are you actually able to calculate the volume of the balloon or the relative volume simply by measuring out with a ruler the diameter of the balloon? Would be also interesting. You take a picture and then you kind of, I don't know, and you calculate the diameter here from here to here. Yeah, or because it's not completely spherical, also from here to here, you take an average. Yeah. So, what cannot be seen? What can be seen in the pictures? Well, with 3 spoons. Okay, that is simply a description of the stuff. Okay, that's fine. It does not have a significant progress. I mean, it's simply a description of what. Carefully, listen carefully. I was here some bubbles. Okay, he's making an attempt here to actually. Describe what he's seeing, but the question is now, how does this answer the research question? Is this in agreement with the research question? So where's the link to the research question here? I don't see this. Then the evaluation. He talks, he evaluates the data. No, he does not. Okay, he basically tells me what is a linear trend line. So evaluation means is to what extent is the data actually allow you to form a conclusion. Okay, this is not fully evaluated. You got to talk about the data. I want to see some numbers in here. Yeah? The conclusion here: the result of my experiment is in agreement with published science. Well, okay, give me the reference. What does published science say? That's the whole thing. It's just I don't know. This is not reference. Yeast is a single-celled fungus. Why do you tell me this? In cellular respiration, here he basically talks about what cell respiration is. This can go into the intro, if you actually think you want to have this, okay? Average balloon volume, so basically it's a summary of the conclusion. But now the question is yeah, that's simply, I'm not saying it's wrong, but I mean, that's basically his conclusion. It's simply a summary of what we actually have. That's not a lot. Yeah. So I need more here. These need to talk. Yeah. Then he talks again about some theory which is not relevant, or at least in the introduction you can have this bubble sounds and intensities of the smell, direct proportion. How do you know this? That's in direct proportion. Did you actually quantify the smell and the bubble sounds? Did you measure the loudness of the sound? So there is issues here. Some strengths here, okay, and weaknesses and improvements. Now, what the problem that I can already see here is that he does mention the weaknesses. He does not really evaluate them, but the amount of sugar was not enough. Okay, then how much sugar are you going to use? So make concrete improvements. So missing concrete improvements, and actually want to see some values here as well. So they're actually, that's not a full evaluation, it's simply a list. So there must be more here and also more here. And the other thing is the following, that you gotta, if you want to evaluate, you gotta tell me to what extent are certain things a problem, to what extent, okay? And this is also not done. And the biggest improvement he totally forgot. And that is basically to actually do the experiment with 0 grams of sugar. Yeah, 0 grams per liter, of course, of sugar. And that is something, that's a control. He missed the control. And he talks about the amount of sugar in bottle 1. What I actually wanted to see is if I were him, where is the graph that I made? No, I'll just redraw it here. If he wants to talk about this enzyme, the enzyme activity curve, it could be that he was simply only measuring here, 3 times. Yeah, he got this here. But what about at very high concentrations? Okay, maybe he should actually say he should go up. He should make more measurements to actually see if the curve levels off after some time. That would be interesting. As a matter of fact, I know that from a certain concentration onwards, it's actually going to go down again because the sugar inhibits respiration, something that the student doesn't know, but that's something that he could have researched and talked about. Yeah, but yeah. So basically, my verdict. This is only a first draft. This is not the final version, he's got some data here. I hoped actually that he would have done a little bit more. Okay, but I think there are some misunderstandings. It is, let's put it this way, if he works on the whole thing and he improves it massively, I actually think it could be a pretty good one, despite the fact that the experiment itself is not the most original one. I'll give it not a bad. I'll give it a medium. Okay. That is basically my verdict on this essay, because there is some stuff here. I think he did understand the basic issues, but also due to the fact that this is only a draft, and he got all of the parts in, and he shows an awareness of the main items. But I think lots of details are still missing, and lots of things have to be improved. If this were a final version, maybe not. Okay, so that's it. All the best, and bye-bye.